Good afternoon, everyone. Thanks for returning to join us for the afternoon session of our day one of the summit. Our first post-lunch speaker is Dr. Scotty Yang from Texas A&M. Take it away, Scotty.
Thank you, Julie. Um, how's it going? Hope everybody's doing well. My name's Scotty Yang. I'm based in Texas A&M now, but before moving to Texas A&M I was based in Virginia Tech, where most of the ants DNA work was conducted. So I still put the Virginia Tech into module logo over there, just to make my former department happy. Okay. So let's talk about, this whole presentation is going to be all about ants and spotlight fly and how we involve ants as our eDNA collector to, help detect and monitor spotlight fly. So I'm going to start with this. Probably you guys are familiar with this show, CSI. This is one of my favorite shows back in the days. They're so popular, popular to a level where, each major city has their own version, CSI Miami, CSI New York, CSI Vegas, stuff like that. My personal favorite is Miami. But anyway, it's because Horatio is kind of cool guy in there. But anyway, so let's assume you guys remember this, or if let's assume you guys actually seen this shows before. You probably still remember the drill, right? So the suspect is gone, and the scene looks clean. But the investigators don't panic. They actually put on their gloves, still, they swap the services, collecting all kinds of trace materials in the crime scene. And they bring back those trace material, trying to amplify DNA. And a lot of the time, they can actually figure out who the suspect is without having to actually see them, right? So I guess that's the power of trace evidence. So when I first became fascinated by with the CSI, not only because I want to be a CSI in the future, CSI investigator in the future, but I was really fascinated by the idea that invisible biological trace could actually tell a story. And that concept did not just stay in the criminal forensics, right? Think about this. If trace DNA can solve crimes, trace DNA should be able to reveal biodiversity or even detecting invasive species because the principle is pretty much the same because organisms shed the DNA persists in the environment, and we detect it through target DNA amplifications. Okay. So with that and all the technology advances in recent years, eDNA approach has actually changed the whole landscape of biodiversity research, especially biomonitoring projects, endangered species surveys, and even invasive species detection. If you want to know the whole transformation that environmental DNA bring into the invasive species detection, you should definitely check into this article right there. It's a great article to kind of summarize the whole transformation. But briefly, invasive species detection has traditionally been very like organism centered, which means that you have to see it and you have to catch it or trap it, confirm it. But we all know when it comes to invasive species, by the time we see them, we're often too late. So what eDNA approach can provide us is allowing us to take actions earlier because eDNA basically, eDNA methods basically allow lower detection thresholds while producing even higher detection sensitivities. Okay. Let me give this one example. There is this outstanding eDNA approach developed specifically for detecting and monitoring the spot fly. And I always tell people, I always tell folks, this is one of those pioneer studies that deliver a critical message to the whole eDNA community, especially for those working on the terrestrial system, which, when we deal with the terrestrial systems, it is essential to identify where the target eDNA aggregates, right? This is because terrestrial eDNA is spatially patchy, uneven, and highly localized. If you just sample randomly, you may end up, searching for a needle in a haystack. In other words, you have a better chance getting a good eDNA signal when you actually dive into the biology of the target species, right? Such as feeding behavior, excretion patterns, or even their spatial distribution, right? So this spotlight fly eDNA study from Lockwood Lab at Rogers University, they actually discovered this spotlight honeydew contains detectable amounts of their own eDNA, of their own DNA. And it drips onto understory vegetation, it coats the tree bark, and it accumulates on the surfaces underneath the feeding sites. Okay. So targeting those potential aggregation points, uh, uh, they designed this spray aggregation and tree rolling methods to collect trace honeydew materials. Not only they look super fun, I mean, I would like to be part of that, giving tree a bath, stuff like that. It looks super fun to me. But more importantly, this method significantly improved the detection efficiency and resolution for spotlight fly. Okay. So let's take a look at the workflow of this eDNA method developed in Rogers University. Okay. So when you go at, if you use a spray aggregation, you actually spray the bark surfaces or any vegetation and you collect the runoff water. Okay. Or in the tree rolling methods, once you give tree a bath with your roller, the roller is actually dipping into a bucket of water. So the trace honeydew material ends up resuspended in the water for downstream processing. Right? And next, you have to filter the water through a membrane, pretty much trapping the DNA molecules on the membrane. And the membrane typically needs a special way to preserve to prevent DNA degradations until the further analysis, which includes DNA extraction and target amplification by PCR or qPCR. I mean, it works well. The whole workflow is awesome, but logistically, think about this. It requires field equipment, water transport, filtration setup, and of course, the sophisticated way to, preserve the membranes. Then I start thinking, what if we're trying to cover a large spatial area? It could be pretty challenging, right, logistically. Like, for example, do we like hit every tree trunk? Do we feed every feeding site, or are we looking for every single honeydew deposit in the environment? So I start asking myself, can we make it a little bit simpler, more cost, labor effective, so making the whole, spotlight and flight detection more scalable? Okay. So based on, what we know so far from the Rocket study, the eDNA methods, we know, we know where the honeydew accumulates. We know those aggregation points, and we know that honeydew actually contains easily detectable DNA of spotlight and fly. So the question we have to ask is, who is already visiting those eDNA aggregation points without us telling to do so, right? There's got to be someone out there, feeding on, those honeydew sugary stuff. I kind of give the clue, it's ants, right? Not sure how many of you have tasted spotlight honeydew. I did. Some, a little bit sweet, a little bit sour. I don't really can describe how that exactly tastes, but I bet ants have known that for very long before we did, right? I mean, simply because ants have a sweet tooth. Not only that, ants have long been associated with a sap-sucking insect or honeydew-producing insects such as aphids, millibugs, scale insects, due to this food for protection mutualistic relationship. For example, in this picture, you see these little ants trying to, is consuming a honeydew droplet from this aphid individual. And in return, ants protect those aphids, right? So what ants do is they will always patrol the tree trunks, inspect if there are sap-sucking insects around, and that's just part of ants' routine life. then you may say, well, spot lanternfly don't really produce honeydew the way aphids or minibugs produce. They actually flick their honeydew away. So there's really not a food for protection mutualistic relationship between ants and spot lanternfly. But in the field, we actually observe ants still going after honeydew, super crazy, like a Friday sale, right? So from a detection standpoint, it really doesn't matter if there is a mutualistic relationship, or it's even better for ants because ants get free honeydew without having to commit effort to protect spot lanternfly. Or perhaps we should be able to be grateful if there is no such a protection relationship, because if ants began actively defending spot lanternfly, we might be in bigger trouble. Okay. So we know ants have sweet tooth, but what other benefits do ants bring to the table if we actually recruit them as our CSI field agents for spot lanternfly detections? We all know ants never stop foraging, and they work just nonstop to cover as much ground as possible to collect environmental sugars, including honeydew. Down here, you see different foraging networks by different ant species, each representing the spatial coverage of a single foraging event by a species. So as you can see, the total spatial coverage can be substantial. So if honeydew droplet fall within anywhere within the network, the probability of ants encounter and collect the material I think it's pretty high, right? Also, the way our CSI field agents handle the eDNA sample is another big plus, for spot lanternfly detections. Um, so ants, they do not immediately digest the liquid food. Instead, they actually store the food in a specialized structure called the crop. Okay. There's the crop. It's in abdomen of an ant. What's really interesting is the digestions in ant primarily occurs in the midgut, okay, which is after the crop. So whatever stored in the crop remains relatively intact for a period of time, before the digestion occurs, which is in the midgut, right? So in other words, the honeydew slash eDNA is not instantly degraded. Instead, it's being carried and retained in ants' belly. So again, from the detection standpoint, this means our CSI field agent, aka ants, are not just collecting the trace evidence, they're also doing a really great job to retain and even preserve the sample they collect. So if we get ants involved, we may not require the same water filtration, specialized preservation steps used in the original spot lanternfly eDNA workflow, which may make the whole scaling up more possible, right? Another big plus, but often gets misunderstood, is do we really have to know which ant exactly collect honeydew originally? That do we have to identify which specific field agent has collected spot lanternfly? The answer is we don't. No. Okay, let's take a step back. Okay, remember the crop, that specialized structure that ants use to store the liquid food? It is often referred to as social stomach. So I think that name's sort of a giveaway because ants store food in the crop to share later with their own colonies. So through this behavior called trophallaxis, foraging workers are able to regurgitate the liquid food and share it with other nestmates in a colony. What does that mean? This means the materials collected by a single ant doesn't restrict within that individual. It gets redistributed across a colony. So detection does not depend on identifying a particular ants that collect the honeydew. Instead, the whole colony actually functions as integrated sampling units. So there's a pretty good chance, you go outside, you collect ants, most of ant individuals in the environment may carry some extent of spot lanternfly honeydew. Okay, so Imagine we have thousands of this our CSI field agents conducting continuous sampling in the field, and this sampling can be very targeted to something sweet and with great spatial coverage. Doesn't that sound awesome? Let me tell you something even more awesome is we don't have to pay them, right? They're free labor, put this way. So with all the benefits, I don't know if I have you guys convinced, ants can serve a scalable CSI agent for spotlight and fly. So let me show you some data that's put ants to work, right? So, but before we dive into the results, I just want to walk you through these two terms, PCR and qPCR. Don't panic. This are, I won't go into detail. These are just a 2D analysis we use, and both are very spot lanternfly specific. They don't cross-react to other plant hoppers or ants. Also, qPCR is a lot more sensitive than PCR, but PCR can be a little bit cheaper than qPCR. I just want to put these two methods right there. I know there's a trade-off, but we can always decide which one we want to use depending on the circumstances, right? And you're gonna hear me mention ants, this ant and that ants being tested in the following slide. I actually mean worker ants, so no other cast of ants being tested. Okay, only worker ants. So the first thing we did is we went to this spot lanternfly confirmed infestations, and we looked for a tree of heavens, and we collect a whole bunch of ants of different species from tree of heavens. Okay, so and turns out all ant species tested positive for spot lanternfly DNA, with PCR producing overall 60% detection rates across all individual ants and more than 80% detection rates using qPCR, which kind of makes sense because qPCR generally has a better sensitivity than the standard PCR. Okay, but I do want to mention here that we actually identify every single ant species to a species level, but we listed as PP here because we don't want to give people the impression that they have to be picky on ants, right? I got asked this question a lot, like, "Hey, Scotty, which ant is the most effective CSI agent?" Put this way, but my answer is based on this data. My answer is every ant can be a hero. Any ants that's environment has pretty good chance to come across the spot lanternfly honeydew. Okay. And next, we want to confirm that spot lanternfly signal is recovered from ants are not simply due to external surveys contamination. As you probably know, when honeydew is abundant in the field in the environment, when ants get to that, they're probably gonna get some honeydew on them. So we design a set of negative controls. So first, in that the same spot lanternfly confirmed site, we collect other insect species, right? But those insect species have very limited interactions with spot lanternfly honeydew. They don't touch that, and we test them for spot lanternfly DNA. We also went to several sites with no prior records of spot lanternfly infestation. I wonder if we're gonna have that sites anymore, but with uh, with the current spreading of spot lanternfly. But anyway, we were able to get some naive ant populations. Okay. Result shows, none of these insects and the naive ants tested positive for spot lanternfly signal. So only those ants collected from the spot lanternfly infested site show detectable DNA signals. So that's pretty straightforward. The signal we recover is something in their belly, not from their external surface. Yeah. And again, and next we're trying to understand how long does that, honeydew slash DNA signal last in ants. So we set up several ant colonies, and we feed them with either spot lanternfly honeydew as the video shows, or fresh spot lanternfly tissues for 24 hours. And after that, we replace it with a pure sugar solution. And then we sample 10 ants every day to test spot fly DNA signal. I mean, here, the reason to include spot fly tissue is that we suppose some of the signal may derive from spot lanternfly crops, right? Because ants are scavengers. They're getting when they encounter insect prey, they're just gonna eat it. So we suspect some of the signals may come from the spot lanternfly crops. Okay, so the image here is the result, which is the gel pictures. You know, maybe it's not very friendly to read, but let me walk you through this. So each band there is a spot fly specific DNA. Okay, and the band intensity you see corresponded to the amount of amplified spot lanternfly DNA. So the thicker, the fatter the bands are, the more spot lanternfly DNA in general. So what we found here is spot lanternfly DNA signal remains pretty much detectable for all through 6 days in those ants feeding on honeydew meal. Okay. But surprisingly, or not surprisingly, but in those ants feeding on spot lanternfly tissue, we can recover very strong signal in the first day, indicating a high spot lanternfly DNA concentration, but the signal declines sharply afterwards. So this is kind of consistent to ant feeding biology, where worker ants are not able to utilize or process solid tissue, but very much competent at retaining liquid food. In other words, our data basically show tissue honeydew can be a good eDNA sources, but DNA from honeydew simply lasts a lot longer in worker ants compared to the spot lanternfly tissue. But when we apply qPCR methods, which we know it's more sensitive, the detection rate that signal can remain detectable up to 2 weeks. So that gives us a 2 weeks window to detect them, which is awesome, right? So now comes what I call the moment of truth. Okay, can this actually work in the field? Or can we actually scale it up? So typically, large scale ant collection is pretty straightforward. As I mentioned before, ants are always foraging, they're always hungry. So if you can just hit them with what they like, they're gonna show up. Okay. So one of the easiest way to do is at the large scale is with transect of lure stations. Where we put piece of cotton ball soaked with a honey solution inside a falcon tube. And what you have to do, just simply lay it down on the ground. You go somewhere else, get a cup of coffee, stuff like that. You come back 20 minutes later. Then you collect ants, you return the ants to the lab for DNA analysis. Then you may ask, how long does it take to set it up? Setting up a transect with 10 lure station with each other spaced by, let's say, 10 meters takes less than 5 minutes. How expensive or how inexpensive it is. Each lure station, is less than 50 cents. So setting up a transect really gonna cost you less than $5. And from the ecology standpoint, with this transect design, we can also look at how far the spotlight DNA signal through the ant foraging networks. Okay, so we manage to identify sites with only single spot identified infestations, and we establish transect radiating outwards from the infestations. Okay, and these sites represent high, medium, low density spot lanternflies, and so we can determine if the detection rate actually corresponded to the infestation intensity. First, we're looking at the high density sites. Each circle represents the lure stations. The black one meaning there are ants, but without any spot lanternfly signal. Yellow one, ants with spot lanternfly signal. And the red one right there is where the spot lanternfly core infestation is. Again, I mean, ants collection pretty successful. 90% of lure station with ants, and roughly 63% of which tested positive for spot lanternfly signal. What's really interesting is Actually, this construction site, this is actually a construction site, and all the tree of heavens with spotlight and fly are actually be behind the fence. Okay, so looks like our CSI field agent were not discouraged. Instead, they still managed to deliver results. So this means, detection of spotlight and flying properties with restrict access is it's possible. Median density, pretty much the same result. We saw 60% of detection rates. But what really surprised us is you can see that spotlight and fly signal pops out at the lure stations located well beyond the core infestation. That means the effective detection range is now limited to the exact location of the spotlight and fly infestations. Okay, if you use the AND methods. Then here comes the low density side. I guess this is what you all probably care about the most. Can we actually, can ants actually do their job picking up the low density? So again, ant collection, big success. All of them have ants. All of the lure stations have ants. Only 47% of them tested positive. Of course, lower than the previous two sites, but understandable because after spending 2 hours of searching of the entire site, we were only able to find six spot lanternfly individuals. But that six spot lanternfly individual actually translate into 47% detection rate, which is pretty good. We need to give our field agents some credits. With their help, intercepting the early stage invasion, becomes possible. So what's next? So we have several ongoing projects here. I mean, I don't Texas doesn't have the spotlight and fly yet, but we still keep working on this. So, but one project I would like to highlight is we are currently developing 100% field spot land and flight detection kits using the ANTS platform. So all you have to do just look for the color change, and equipment wise, you don't even need this sophisticated PCR qPCR machine. All you need is probably a heat block or even a thermos that can heat the liquid up. And my student Peter Ship, he's based in the Virginia Tech, and he's the one who is working on this project. But ultimately, I would like to push this ants DNA platform to be a novel agricultural biosurveillance tool, specifically targeting those honeydew producing insects, and many of them are pretty notorious invasive species. Yep, that's all my presentation. That's a little workflow of our ant DNA. Thank you. With that, happy to take questions.
That was a great presentation. Scotty, you have some uh, questions in the chat. I'll read one to you
Okay,
from from Michael Formicelli. Is it possible to locate a population based on the signal you get from the ants collected from different parts of the area? I am
perfect.
thinking
Yeah.
based on your graphic showing the range of the ants and trying to find it in densely populated areas.
Yes, so that's one of our ongoing project. We are trying to come up with model to predict where the spot lanternfly infestation, is based on the spot lanternfly DNA across the transect. So I mean, our current model shows the prediction is possible, but we need to do more verifications this summer.
That's fantastic. And then one more. So it just came in. Could this method be adapted, at least in part, for citizen scientists?
Perfect. That's exactly our kind of our goal, just get everybody involved. Once this field 100% field kits is ready, we definitely would love to do some sort of a citizen science, or we can just have, citizen scientists to collect a sample, send a sample to us, and then we can run the molecular stuff.
That is fantastic.
Thank you.
Thank you so much.